Distinguished member of Reproductive Health Supplies Coalition, good morning and good afternoon. I'm Dr. Wei Jin from BioPerfectus. As a Chinese manufacturer, BioPerfectus is honored to have this opportunity to be a new member of RHSC and deliver this keynote speech at this membership meeting. Standing as a leader in the molecular diagnostic industry, BioPerfectus specializes in infectious diseases and women and children health. Founded in 2010, BioPerfectus has consistently focused on R&D, manufacturing, and the distribution of IVD and instrument. We have developed a range of over 600 products concerning reproductive health testing, such as HPV, vaginal secretion detection, STI, GBS, and so on. BioPerfectus is also an advocate, industry leader of vaginal microecology. So far, BioPerfectus has more than 1,000 employees, and in 2022, the annual revenue has reached 5.5 billion yuan. On December the 5th of the 2019, BioPerfectus is landed on the Shanghai Stock Exchange as the first molecular diagnostics company. Since establishment, we have not only focused on domestic construction, but have also actively expanded to overseas market in order to create a global brand with international competitiveness. So far, our product has penetrated to more than 100 countries and regions. Branches have been launched in Hong Kong, UK, and the United States. Driven by the mission for a healthier future, BioFactors employs precise, effective, and streamlined diagnostic techniques. The aim is to pioneer advanced early screening and diagnostic methods for infectious disease and major health challenges, thereby improving the quality of life and promoting global health. In the post-COVID-19 era, the public is increasingly concerned about their health, particularly in reproductive health. Cervical cancer, which is ranked at the fourth deadliest cancer among women by the International Agency for Research and Cancer, has become a public concern. Over 600,000 new cases, more than half of which result in death each year. Both the incidence and the mortality rate are high enough to indicate that this cancer severely threatens women's health. Cervical cancer is a preventable disease from the progression of HPV infection to cervical cancer. It can also be cured if detected earlier and treated appropriately. HPV genotype and the viral load are very important factors driving the cervical cancer contractions. In order to induce the global mortality from cervical cancer through implementing effective intervention at a different stage, WHO has launched a global strategy to accelerate the elimination of cervical cancer on November the 17th of 2020. It marked the first time that 194 countries have committed to eliminating cancer. Cervical cancer is largely preventable through both vaccination and screening for precursor lesions. With appropriate follow-up and treatment, as a secondary objective, screening may lead to leading detection of cancer, which may allow for earlier treatment and a reduction in the risk for death from this cancer. HPV DNA testing has been recommended by WHO as a primary screening method for cervical cancer. For technology, HPV testing method has undergone a process of development. Firstly, a hybrid capture technique was used. One uh, cannot distinguish the specific types. With the progress of clinical research, more and more evidence show that the importance of genotyping. For example, HPV 16 and 18 genotypes were the first recognized with strong carcinogenicity. So products that can distinguish 16 and 18 genotypes appeared. In the last 5 to 10 years, more studies have been conducted on the correlation between the HPV infection and the cervical cancer. Gradually, except HPV 16 and 18, other genotypes like the 31, 33, 52, 58 had also occupied a high proportion in the cervical cancer. Therefore, in line with the clinical trends, product can detect the extended genotype appeared in the market. In addition, HPV viral load is also thought to play an important role in the development of cervical cancer. In China, HPV detection technologies are on par with the imported product. In 2015, the CFDA approved the BioPerfectus HPV-21 genotyping and quantitative detection kit, which can perform both genotyping and normalize the viral load determination. From the perspective of the application, HPV detection technology developed from no genotyping to extended genotyping, from qualitative to quantitative. In 2015, BioPerfectus became the first and to date the only company to provide HPV detection kits for both 21 genotyping and the normalized viral load determination by multifluorescence quantitative PCR in separate tubes. As we know, the main challenge in achieving accurate quantification is sampling difference, which can lead to a varying number of cells in the sample, making it difficult to establish standardized measurement and ultimately achieve accurate quantification. In this regard, BioFactors has made an important breakthrough. First, they determined the viral load of each HPV type using the multiplex qPCR. Then they measured the number of cells in the sample by detecting the single copy gene present, allowing them to report the viral load of different HPV types in the same cell. That is, the number of HPV viral load per 10,000 cells is reported in the basis of achieving normalized quantification. Measuring cell number by detecting single copy gene is the key technology to achieve quantitative result. The single copy gene is the top 3 gene, which encodes human topoisomerase 3. It is published in PNAS in 1996, which reported that this gene is a reliable single copy gene in human cells. As shown in the figure, we employ two different methods to validate the reliability of using top 3 single copy gene for cell number calculation. Firstly, for the same sample, the number of cells is calculated by using the classical cell micro-mounting method. Secondly, we're using real-time PCR to quantitative the number of cells in the sample through the standard curve. It is found that the quantity of value of the two methods are the same, and the deviation range is less than 0.1 log. In this way, we could confirm the reliability of using top 3 single copy gene to calculate the number of cells. This is in line with the European guideline, which emphasizes the need to adjust HPV quantification based on the number of cells per sample. BioPerfectus is perfectly in line with this consensus direction. In clinical study, more than 8,000 cases was compared between BioPerfectus and the Roche. The conclusion is that the BMRT are the same sensitivity as COBUS, but the BMRT has the highest specificity with COBUS for primary screening of cervical cancer. The HPV viral load with typing as the primary screening triage protocol for HPV positive can achieve the same sensitivity as the HPV 16 and 18 combined cytology triage protocol, and the BMRT can complete the primary screening and the triage in one test, not requiring cytology. On this basis, the FCLSC cohort study led by Professor Sun and his team screening about 120,000 women to evaluate the role of HPV genotype and the viral load in predicting the risk of high-grade cervical lesions. The study has found that there's a positive correlation between the viral load of HPV-16 and other four genotypes with the severity of the cervical cancer. The optimal viral load for specific genotype can be obtained as a clinical cutoff value for cervical cancer screening, which means HPV genotyping combined the viral load can be used as a triage tool for screening HPV positive patient. Here's a summary of the clinical value of HPV viral load, which is mainly seen in monitoring HPV infection, predicting the progression or the outcome of the infection, assisting in triage, and observing the effectiveness of treatment. For this technology, it has been already applied in China for nearly 10 years, and 16 papers have been published. In the clinical end, recently HPV genotyping and quantitative detection was also introduced to the international community. While in order to accelerate the elimination of global cervical cancer. Besides in China, Perfectus Mobiles helped China's two cancer screening program with HPV 21 genotyping and quantitative detection screening strategy. With BioPerfectus cervical cancer screening solution, it helps transform nucleic acid detection capacity in countries and promote screening coverage. In addition, in 2015, the CFDA issued a new guideline for the HPV region. The new guideline requires the prospective clinical study with follow-up. BioPerfectus was among the first tier of company to follow this guideline and initiate the ANITA study based on more than 30,000 women in China for both the high-risk screening and HPV genotyping test kit. This study lasted for 4 years, from 2017 to 2020, and was conducted on 3 research centers and more than 13 clinical trial sites. The research report was about to publish soon. In the process of product development, BioPerfectus has always attached importance to the academic exchange and the research. We not only produce high-quality product, but also strive to become a leading company in order to achieve our goal to be one of the top global IVD suppliers. From China to the world, embrace the world with health. BioPerfectus contributes every effort to the health of all mankind. Thank you all for your listening. Thank you.